Good morning. My name is Meg Deloff, and I work for USGS PNAMP. Thanks for joining us for the October PNAMP Fish Monitoring Work Group meeting. A couple of housekeeping tips before we get started. Please mute yourself by clicking the microphone icon on your Teams platform. If you're on the phone, use *6. Also, please turn off your camera if you're not speaking. Sometimes it helps with people's bandwidth or your own bandwidth. If you don't see the toolbar, try kind of moving your mouse around and hopefully it should pop up. You should see something similar to what's on my screen, depending on whether you're using your browser or your desktop version of Teams. If you have any questions or feedback, please use the chat function or raise your hand and unmute yourself. We can mute you, but we cannot unmute you. So you have to do that on your end. We're recording the meeting today. These are typically available publicly on the YouTube channel. But, you know, sometimes people's presentations are a little bit, haven't been vetted yet. So sometimes these don't show up on YouTube, but, we can find out ways to get you information if you do want something. But Nat graciously has said it's fine to publish this one for this meeting. So I will send a note with the follow-up email, just giving you information on how to link to the YouTube. If you're having trouble with chat, here's a couple of troubleshooting tips. Try leaving the meeting and joining again. Use a different browser. Edge usually works better for Teams than Chrome, as Nat demonstrated for us this morning. Or if you use the desktop version instead of the browser or vice versa, sometimes that kicks Teams into working. If you don't have access to the chat, please feel free to unmute yourself and comment or ask questions. It's not a big deal. Quick reminder that PNAMP commits to fostering an open and welcoming environment free from harassment, so please remain respectful. I would like to acknowledge all of the Fish Monitoring Work Group leadership. Names are associated with the tasks they have or currently lead. Core team, if you guys could turn your cameras on and introduce yourselves, that would be great.
Do you want me to go first?
Yeah, go ahead.
I'm Nancy Leonard. I'm with Pacific State Marine Fisheries Commission, and I serve on the core team.
Thanks, Nancy.
I'm Jen Bayer with USGS and PNAMP, and I help with the core team as well.
I see you out there, Casey.
I'm out here. I'm Casey Blazner. I'm with the Oregon Department of Fish and Wildlife with the East Region Fish Research Office.
And also on our core team is Laura Erickson. She's with PSMFC, and Marika Dobos, who's with Idaho Fish and Game. Marika's not available. She's out in the field, I believe, for the rest of October. And also Russell Scranton. I'm not sure if he's in yet, but he's part of EPA. And so everybody, thank you, core team members. You guys can go ahead and turn your cameras off. Now it's participants' turn. You guys don't have to worry about turning on your mics or your cameras. Just simply open up the chat and introduce yourself with name and organization. This helps acquaint you with the chat function and helps us keep track of participation. And then while you guys are doing that, I'm just going to quickly cover today's agenda. After I wrap up with this, we'll kick off the meeting with some updates on the current tasks and a completed task and a task that's kind of in progress right now. And then next, Nat Scholz will give us an update on NOAA Fisheries stormwater science in the Puget Sound, followed by Q&A, and then we'll close the meeting with just some next steps. And with that, here we go. Leads for each current task will give an update and progress, and then I'll pause briefly after each update and ask questions or kind of look for comments. If you have a question after we've moved past a task, type it in the chat and I can kind of circle back and take a look at that and read them, or Jen can see that for me and just kind of read them off if you're too shy to unmute yourself. And with that, I will hand this over to Morgan Bond with NOAA.
Good morning. So our task carrying capacity standards creation has been going for a while now. The real focus of this is to try to sort of put some clarity to a bunch of different models and approaches that people have for estimating carrying capacity for particularly salmonids, but stream fishes. And there are so many different sort of resources that people are using to do this type of work that we're putting together a document that really outlines those different approaches and then makes recommendations for maybe what approaches should be used under what circumstances and how to interpret and really properly use those different approaches. So including both what information goes into that as well as what the kinds of outputs are. So we put together this document. There's about a dozen co-authors or so on this. So it's a pretty big effort to put together information for all these approaches. Got about 60 pages so far, and we're working on putting together, I think, some tables and figures that'll be really, I think, useful to help people be able to navigate this kind of complex issue. And so, yeah, we're just in this writing and editing process. And I think that's kind of our update for right now.
Awesome. Thanks, Morgan. Does anyone have any questions for Morgan? Or I think Mr. Copeland, if you're out there too, Tim can help ask or answer questions. And also just feel free to type it in the chat if you're feeling shy. All right, I'm gonna keep this train moving. You guys can obviously circle back later and ask questions if you come up with something in a little bit. Next, I'm going to talk about kind of the next couple of tasks. There we go. Sorry, guys. If you recall, we hosted a meeting where Dan Isaac gave a demo of the fish distribution analysis tool. A call for data was sent out earlier this year, and that data was sent to Dan Isaac and Gwen Chandler with U.S. Forest Service. They've been processing the data, and in working through this effort, will aid in identifying recommendations for metadata input formats to improve the FDAT tool. And then we're probably going to review and update the juvenile density vocabulary spreadsheet based on that feedback. So it just makes a bit more of a streamlined process for inputting data. Does anybody have any questions? And Jen, I can't see chat, so if you want to read them, if they pop in, let me know. All right. And Casey,
There
if
is,
you guys.
wait, there is Ian says, isn't it the fish density analysis tool?
Fish?
I think you're right, Ian. Sorry, just a
Oh, sorry.
typo
Yeah,
there.
no. thank
FDAT,
you.
affectionately known
Totally.
as FDAT.
Yeah.
Thanks, Ian.
Thanks, Ian. All right. So next one, rotary screw trap operation. In June, we convened regional experts and gathered input on the StreamNet rotary screw trap dashboard that Nancy will be talking about in just a second. We'll continue to work with StreamNet and regional partners to integrate controlled vocabulary and standard fields into a common trends database. And Nancy, if you want to add anything, that'd be great.
Yeah, so a tool or a database dashboard / outreach tool that we've been working on that relates to this work group is a rotary screw trap dashboard that we've been building on Esri. We started off sort of like a prototype to find out how we could bring more easily content from different existing databases together, so it's easier for folks to be able to learn about associated information for any rotary screw trap that's funded by BPA. We developed a prototype layout of how we wanted to bring together information from monitoringresources.org, from BPA's CB Fish Pisces data system, and from the StreamNet fish facility map tool that we have, as well as the StreamNet data systems that we have that contain data collected by rotary screw trap. We mocked up the dashboard of where we would put things and what type of content we would be displaying on an Esri dashboard when people would click a certain screw trap, and what would be popping up, as well as what type of additional information we would make available on a different site related to those individual rotary screw traps shown on the Esri mapper. We shared that type of content and how we wanted to lay it out with the fish monitoring work group earlier this year. We got some feedback from them as to how we should organize that content. We updated that dashboard, and then we reached out and received input from the StreamNet steering committee, which they provided input on that mock-up back during their September meeting, and we are refining that layout, and we'll be sharing that with the executive committee, the StreamNet executive committee, in November. Once we get thumbs up on how we're displaying what content on the Esri dashboard and how, just as the layout and how it looks and feels, the functionality, then we will come back to the fish monitoring work group to start asking for the biologist that has expertise in terms of this information related to rotary screw traps, so that we can actually start going through it and making sure we have all the screw traps that should be displayed and that the information we are associating with those screw traps are accurate. So hopefully by early next year, you'll be hearing from Meg that we're ready to start actually diving in and looking at the actual accuracy of the content so that we can move into phase 2 of the dashboard. That's it, Meg.
Awesome. That was wonderful, Nancy. Does anybody have any questions for Nancy? All right. Next task is pit array and related pit tag analysis. Pit array and related pit tag analysis is focused on documenting and recommending improvements to data management and analytical tools for pit tag array data. We've been coordinating with Pitagus to participate in discussions surrounding pit tag array data at their upcoming workshop. The last time the workshop was held was in 2015. We feel like this is a great opportunity to ask questions and gather feedback on the needs of the regional pit tag community. The workshop is scheduled for January 30th through for February 1st. Registration is currently open, so Jen's going to drop that link for you guys. It's a link to the workshop page, but the registration link is also on that page. So in case you want to get on top of that, it should be a really great workshop. Anybody have any questions on that one? All right, excellent. This task is in development. In April, Coordinated Assessments Partnership convened the 2023 CAP workshop. Fish Monitoring Work Group is coordinating with CAP to support needs identified at the workshop. Some of the priorities identified was gathering the community to review definitions on the DESs to confirm consistent use. Fish Monitoring Work Group is a great platform to use to connect regional experts and provide input on the review process. This will be as needed and spaced out over time as to not overwhelm you guys. And we are still working on a schedule for specific tasks. And so we're still in development. There's not a whole lot of information quite yet. Jen, did you want to add anything to that?
No, I think we're thinking that the review of the definitions and terms business will start, I don't know, like Nancy, do we say we're thinking like late spring, early summer? So to where we actually want to engage all you experts to help make sure we're all saying the same thing, apples to apples. So what we're doing right now is figuring out a good process to do that efficiently, and so we can track it through time into the future. So we'll, it's coming. It's probably going to be, it'll be a very important task, and we're trying to do it as efficiently as possible. So I don't really know how much time, but it probably won't launch until the spring at the soonest.
Awesome, thanks, Jen. Does anyone have any questions on the CAP task? All right. Next up, we completed the fish management units and boundaries and attributes task towards the end of July, beginning of August. The task we helped facilitate for this task in particular. We helped facilitate the recommendations for defining the fish management units and attributes for StreamNet tools. Nancy can give a little bit of additional detail on this, and Jen is going to drop a link to the recommendations document in case you want to take a peek at that. It's housed on the pnamp.org site. Nancy, did you want to add anything about that?
Sure. So this was really a great step forward for us to finally have good input on how we can go about developing management unit boundaries that are less specific than the individual population or where fish are actually located in the system. So it's a slightly more general set of boundaries that we can use when we're developing web-based query tools that rely on map interface. So we received this set of recommendation from the Fish Monitoring Work Group. And during the September StreamNet Steering Committee, we reviewed it with the steering committee folks to get additional input on the recommendation that we received from the Fish Monitoring Work Group. And our next steps now is we're going to take the input we received from our StreamNet Steering Committee members and basically use that to package up the recommendation received from the Fish Monitoring Work Group so that we can deliver that for review by our StreamNet Executive Committee in November. And then if all goes well, we'll come back and give an update to the Fish Monitoring Work Group to describe how we will be implementing these recommendations.
All right.
Back to you.
Excellent. Yeah. Thanks, Nancy. That was great. Does anyone have any questions about this task? And Jen, you're on chat. And then also, this is the last task slide before we do Nat's presentation. So if you guys have any questions that you thought of, feel free to unmute yourself and ask questions about any of the tasks if we kind of ran through that too fast. I'll just give it just a minute.
I'm not seeing anything in the chat, Meg. Oh,
Okay.
wait. And Ian, no, Casey dropped a link to the FDAT, confirming that no, it actually is the data analysis tool. Thank you. ODFW folks, thank you very much, because we actually now have a link to the publication of the first version. And what we're trying to do is help Dan improve that. So thank you to all of you who provided data when we did the data call earlier this year, because that's what Forest Service research team is doing right now, is using your data to update the model, and they will come back to us with some improvements later.
Perfect. Thanks, Casey, and thanks, Jen, for elaborating. All right, and with that, I am honored to introduce Nat Scholz as, oh my goodness, sorry guys, I have a mushy mouth today. I'm honored to introduce Nat Scholz, an ecotoxicology program manager at NOAA Fisheries in the Northwest Fish Science Center. Take it away, Nat. And I am going to stop sharing my screen.
Good morning, everybody. So again, just trying to get the share screen to work out. Of course, we just did this while we're setting up.
Yeah, no, it's the nature of this. Yeah, it's totally fine.
See if that does it.
All right, yep.
Yeah, there's a lag time on my browser, so I'm actually not seeing the slides yet.
Okay.
But you guys can see them, huh?
Yeah, it's still showing your kind of desktop version.
Let me go to full screen mode. How's that?
Perfect.
Okay, that's awesome. Good morning, everybody. Happy to be here. Thanks for having me come and give an informal overview of our stormwater science, nominally focused on Puget Sound. My name is Nat. I head up the ecotoxicology program at the NOAA's Northwest Fisheries Science Center. So I'm at the same research organization that Morgan is, and we actually are in different divisions and run in somewhat different circles. So this is kind of a cool connection. What I'm going to try to do today is give you guys an overview of the science that we're working on around environmental health of salmon and other priority species for NOAA in the context of stormwater and coastal development and urbanization. So a lot of the, I'm going to bounce all over the planet with some of this science, but a lot of our stormwater science is really focused in Puget Sound with an eye towards other geographic areas up and down the West Coast. And I'm going to talk a little bit about work on tire chemicals and 6-PPD, and also a lot of work on developing new markers and new indicators of fish health that can be used for monitoring in fish habitat, so in the field. And so again, there's going to be some background information and then some specifics on emerging chemicals of concern, and then essentially a lot of new science at the very end with a real strong focus on physiology. And I just want to acknowledge an extended team at the Northwest Science Center. This is comprising the work within the ecotoxicology program that I lead, and then also the analytical chemistry program. And so again, it's a large army of people. Megan, if I move my cursor around, can you see the arrow? Probably very slow.
I don't see it.
Okay, well, the guy in the bottom right, John Incardona, is leading a lot of the cardiovascular work that I'm going to talk about today, so I just wanted to point him out as well. And we have a very large network of collaborators across the region and across the country as well. On the NOAA side, this is kind of the operating framework in which we do science. We have a separation of church and state within fisheries. So the management side of the agency is responsible for making the decisions around natural resources, including salmonids and forage fish that I'll talk about today. They invariably are operating under major federal statutes. Dominant ones for the conversation today are going to be the Endangered Species Act and the Oil Pollution Act. And then, information gaps come up, or, major uncertainties arise, and this adaptive management process. And the science centers around the country typically are providing new information and targeted applied science to answer kind of these key management questions. And ideally, this is this infinite rotational loop of adaptive management and science. And again, the goal for today is really targeted science to better understand habitat conditions as they relate to water quality in areas that provide critical habitat for ESA-listed salmon and food webs, including marine forage fish. The topic is coastal development and urban growth, familiar to pretty much everybody around here, but essentially what this reflects is a process of land use change, so urbanization and the gradual replacement of forested or agricultural land or rural land with more and more pavement and impervious surfaces. As that happens on a death by 1,000 cuts scale, you gradually are converting the watershed to more and more hard surfaces where the rain can't infiltrate and runs off, and it runs off streets and roads and roofs. And when it does that, it carries a ton of pollutants into nearby waterways. And so this is called the urban stream syndrome. It is a combination of physical and chemical and biological habitat factors, but anywhere in the world where you have coastal development, as we do here on the West Coast, you see this gradual decline in biological integrity within urban watersheds. And this has been studied for years. These papers that are on the right, are 20 years old, and this was a big deal 20 years ago. And urbanization, kind of like climate change, was on everybody's radar screen 20 years ago and more. And again, with patterns that we're seeing of people moving to the coast of the United States and elsewhere, we're seeing this intense pressure on coastal watersheds and watershed health as a function of non-point source pollution. And a lot of that is related to the wash off of a lot of these contaminants in urban stormwater. So again, we have a lot of mission spaces within fisheries. I'm going to bounce back and forth between predominantly salmon and forage fish today. But in the context of salmon and the endangered species work that we do, the targeted research is really targeting 3 different management domains. The first one is recovery. And this is really science to better understand what the limiting factors are for salmon population recovery at the basin scale. So this is what we call evolutionarily significant units. And ultimately, this is a very large level analysis of trying to understand how water quality is a limiting factor for, basically highly migratory salmon that are moving through these urban and non-urban watersheds as, across different stages of their life history. So we do a lot of work on trying to understand the aggregate population scale level impacts in the context of recovery. Restoration is really all the money that's going through the agency right now and other agencies to try to improve salmon habitat. And this is really focused on fish passage projects in part. And so we're really trying to understand and make sure that water quality conditions from stormwater and other sources are not critically undermining a lot of the ongoing physical habitat investments that are really just focused on migration barriers and access, right? Because ultimately we're opening up habitat in upstream areas that are highly urbanized or going to be urbanized over the next year, to 10 years to 20 years. And so ultimately understanding how the hydrology and flow of water carrying contaminants comes into these restoration sites is really important to make sure that we hit the recovery goals that we want. And then the third category is regulation. And this is intrinsic insider baseball for NOAA and U.S. Fish and Wildlife Service. So ultimately, this is science to help our managers make decisions when they're reviewing either very specific projects like a bridge replacement or a highway project, or batches of similar projects in a programmatic consultation. And so basically the science is in these domains for recovery and restoration and regulation. All three of these domains have a monitoring component, right? So there's effectiveness monitoring, there's status and trends monitoring, there's all kinds of monitoring. So, and this is a monitoring, savvy community. So you guys see the places where it plugs in. Puget Sound, for stormwater, we've been focusing for the last couple of decades, really since the early 2000s, on coho, the sentinel species for stormwater impacts. There's a good reason for that, and that is the overwintering life history. Coho tend to spend more time in small streams in areas where people like to live. And you see on the left, there's kind of a gross indication of urbanization in Puget Sound, and you can see how that overlays with where we have coho in space and time. And we've been essentially using coho as ambassadors, for not just, salmon recovery in Puget Sound, but also the communities that they depend upon. So the integrity of these aquatic food webs in marine waters, the integrity of marine food webs, including forage fish, for Chinook and the like. But for coho in particular, a lot of our science is really looking at, exposures that happen during rain events that are distributed throughout lowland urbanizing watersheds. And again, the talk today is really is centered on coho, but again, we're mindful of recovery planning and the need for this kind of stormwater science throughout the entire range of all of our ESA listed salmonids on the West Coast. And again, a lot of our stormwater science is particularly focusing on coho, and you can see that in the C's to the left. But we also are doing a lot of work increasingly on steelhead. And so between coho and steelhead, we're into recovery domains that span almost all of the salmonid range in the West Coast right now. I should also mention and give a shout out to the Puget Sound Federal Task Force. So in our local area, we have a coordinated effort that's being led by EPA as part of the National Estuary Program. This is a major source of funding to NOAA and other federal agencies to tackle these really complicated problems in a coordinated way. And so ultimately, stormwater is one of the major issues. There are several others, obviously, in Puget Sound. But again, we are now going into our third of 4-year agreement with EPA to try to better understand what the threats are related to stormwater. And then ultimately, we've been doing a lot of work on solutions as well. So this is probably familiar to most of you. We've been working for a couple of decades on this phenomena called the urban mortality syndrome. And this was first discovered actually at a restoration site in West Seattle. This is Longfellow Creek. And examples of fish passage projects in the 1990s that the city of Seattle was investing in. And ultimately, with these culvert replacements, they hired what is now the Wild Fish Conservancy to come out and do stream surveys. And they were finding that the fish were coming back into the newly available habitat, but behaving very strangely, like you're seeing here. So again, in fall of 2000, it starts raining, the coho are coming back into these coastal watersheds, and within an hour or two showing this distress syndrome, which is, we call it the pre-spawn mortality syndrome. And essentially you see these fish that are swimming at the surface and gaping. It's been happening every year in every urban stream where coho are coming back from California to Canada. So this is a widespread phenomena, and we were working on it for a solid 15 years as a massive forensic investigation, trying to figure out exactly what the causal factors are, and then what the consequences were going to be for the salmon recovery mission for NOAA. And again, I just want to say there were two major factors that were limiting this line of research in the 2000s. One was that we lacked the analytical technology to be able to break urban stormwater runoff into its component parts. If you collect a water sample, up until the last few years, we really haven't been able to tell you how many chemicals are likely to be in highway runoff. And it's only with this non-directed time-of-flight mass spectrometry Again, this work being pioneered by Ed Kolodja's group and his lab at the University of Washington at Tacoma, Center for Urban Waters. So essentially with time-of-flight mass spectrometry, you can start to break cars, as we have done, into their components from tires or from windshield wiper fluid or motor oil or whatever. And then do parallel analyses of what's in highway runoff, and then also do parallel analyses of what's in salmon streams, right? And so this, what you see on the top there is a heat map. And it turns out there's thousands of compounds, chemicals that are in urban runoff, most of which we have no idea what they are. And so unless you have an analytical standard, you're seeing a peak on a chromatogram of a certain size, and how many nitrogens or carbons or whatever, but you don't know necessarily what that chemical is unless you have a standard. And so what this initial work for us with this collaboration with UW allowed us to do is to actually sketch out the full size of the water quality picture, how many different factors are potentially in stormwater runoff. And you can, if those of you that do habitat work for salmon, that are used to thinking about habitat in the sense of like water quality factors like temperature or dissolved oxygen, each of those is a standalone habitat factor. We just added several thousand new habitat factors to salmon, to the salmon habitat picture in urbanized areas. And so it really makes this whole issue about what is salmon habitat and what is the role of water quality incredibly complicated. But in so doing, we also needed to go through with a higher throughput screen that would allow us to go through this mixture and figure out, precisely what was the smoking gun. So we were able to narrow down the size of the haystack. And Zhenyu on the right was the postdoc that was on the project that eventually discovered the smoking gun, which is 6-PPD, which is a tire additive. It's an anti-ozonant, and it's the derivative, the abiotic transformation product, this quinone, that is so incredibly toxic to salmon. It's not just adults, it's juveniles. So a note about that, as part of the Puget Sound Federal Task Force, we've had a shared goal in 3 category areas over the past 5 years. One of them is really just trying to understand the nature and the threats of stormwater to salmon at Puget Sound. We've also been doing a lot of work on green infrastructure so that we know the water is toxic. And so what is going to potentially clean it up? What kind of mitigation strategies are going to be available to local communities, local governments, everybody? Stormwater is a people problem, right? So it's generated by people driving their cars and just doing everything in their daily activities. And so the solutions are also going to be at an individual people type level, neighborhood by neighborhood, installation by installation. And so we've been doing a lot of work on green infrastructure, and then there's a massive science communication and outreach component of this as well. And again, over the past 5 years, with the discovery of this tire chemical that is incredibly toxic to coho, there's been a lot of focus on what this is and essentially how much of it is out in the environment and where it's coming from. And there's all kinds of issues about tire particles being, long-term releasers of these chemicals, and then they get into streams and they get resuspended during storms. And so there's a lot of questions about environmental fate and transport. As I mentioned, we've also been publishing a lot of papers on green infrastructure, working with our partners at Washington State University, Jen McIntyre and company. So really trying to figure out what are some cost-effective and simple treatment methods that can remove this. And in soil columns, the kind of technologies that you see in simple rain gardens actually work really well. And we've shown that they can protect salmon just by taking the really, really toxic runoff from the highway right in front of our science center, the 520 bridge. And you filter that through these soil columns, and essentially the salmon and receiving waters survive. And then again, science outreach and education. So the rest of this talk is not actually going to focus on 6-PPD. We and many others are working on this now, but we're not to the point right now that we actually have indicators of fish health that are going to be operational in the field. And the reason for that is that 6-PPD quinone is one of the most toxic chemicals now known in my field in ecotoxicology in terms of its lethal impacts on salmon. So essentially, we've been doing monitoring for this phenomena for 6-PPD for 20 years. And the endpoint that we're looking at is dead fish, right? Because ultimately, what you're finding is prematurely deceased spawners in your urbanizing habitats. And if you cut the females open, they're full of eggs. So that's still the standard. As we're working out new methods analytically to measure these compounds in fish tissues, and then we're really specifically looking at methods to understand sublethal effects in species like steelhead. So this is going to be the topic of the future kind of update for you guys. What I want to do is go back to familiar contaminants in stormwater and talk about progress that we've been making there, because again, solving the 6-PPD quinine problem is not going to solve the stormwater problem. There are so many other known and unknown bad actors in stormwater that we're working on. So it'd be familiar for our work on metals, for example, things like PCBs. But I'm going to spend the rest of this time talking about PAHs. PAHs are fossil fuel derived polycyclic aromatic hydrocarbons. They are ubiquitous. They are a major human health concern as well as an environmental health concern. They are dominant in oil spills. They're dominant in stormwater runoff. They're dominant in industrial polluted sites like Superfund sites in the lower Duwamish or Portland Harbor. They're a major source of atmospheric deposition. Every time you have an inversion and there's smog around Puget Sound and then it rains and the air clears up, that's because all the pHs went into the water and then went into Puget Sound. So ultimately, these things have been known for a very, very long time, but they are also a major source of scientific uncertainty. And so I want to talk about that a little bit because we're developing new tools for addressing these in monitoring type applications in the field. So you'll see in the slides that follow me switching back and forth between stormwater and oil spills, and that's because we work on both and the compounds are the same and what they do to fish is very much the same. And so I'm going to use examples that are illustrative that might be pulled from one oil spill or another, but we've worked on many oil spills around the world. My group has had two dominant ones. One is 1989 Exxon Valdez, and the other is the Deepwater Horizon spill. These two spills have defined two generations of researchers at the Northwest Fisheries Science Center. The Exxon Valdez spill for the generation that preceded me, and then my group worked extensively on the 2010 Deepwater Horizon. And our focus area there was really the bluefin tuna and other large pelagic predators that were spawning in the offshore areas of the Northern Gulf. So every time there's an oil spill, if you work on ecotoxicology, there's usually a massive federal investment in science. And so we kind of go through this boom bust cycle with our research funding to do work on PAHs, depending on whether or not there's a major oil spill. So luckily or not, depending on how cynical you are, the Deepwater Horizon oil spill investments, which were unprecedented in the science of what those compounds do to fish, was a huge boost. And we're bringing all of those new tools and all those new insights from the Gulf of Mexico back to Puget Sound and the West Coast and trying to apply them in a stormwater context, again, same compounds, in a way that can help us manage salmon recovery. And so again, just because we're focusing on a subset of compounds in stormwater doesn't really make the picture a lot simpler. There are still hundreds of PAHs, and they vary as a function of molecular weight, which is the number of rings each compound has. They go from naphthalenes in the purple on the left, the 2-ring compounds, to 3-ring compounds, to 4-ring compounds, and 5-ring compounds. And those green compounds that I've circled, the phenanthrenes, you have P0, that's the parent phenanthrene, and then you have derivatives with different methyl groups attached. So the same compound, depending on where you have the methyl groups attached, can vary structurally and have very different effects on fish. And so this is, even though this looks like a simple 2-dimensional chromatogram, it's actually a 3-dimensional mixture. And so I'm going to spend most of my time talking about phenanthrene, these tricyclic compounds, as a focus area because they do most of the damage to fish in the work that we've done. So super complicated, but our motto, even though the chemistry and the environmental exposure issues are very challenging, is always to let the fish tell us what's going on, right? So we are big believers in letting the biology and the physiology and the development of the fish guide us. And again, the focus on early life stages is really important because a lot of our oil spill science was focused on spawning habitats. So in Prince William Sound, it was pink salmon and herring that were spawning in near-shore oiled areas. So they were the focal species for the injury assessment. Gulf of Mexico, as I mentioned, the bluefin tuna were spawning in the upper surface waters. And so a lot of our focus on oil and pH toxicity is at the early life stage. And what you can see is this canonical impact where the normal pink salmon fry with a yolk, this is a yolk sac larvae, to the left. And then you see this fluid accumulation around the yolk sac area and the pericardial area. That's very characteristic. In herring, it looks a little bit different. But again, at the bottom here, you can see around the heart, you can see this big accumulation of clear fluid, and it's pushing on the yolk mass, the anterior end of the yolk mass. And so what these effects reflect is impacts on the heart. And so this is a form of cardiotoxicity. It looks different in different species in terms of where the fluid accumulates, but that is actually more just a feature of the anatomy of different species and kind of where the fluid goes when the heart stops doing its job. And again, as I mentioned, we've been working on oil spills all around the world for a long time, and also on model species like zebrafish. And so we see this consistent syndrome of heart failure as a function of pH exposures in all these different fish. And the arrows are just pointing to that, what I alluded to earlier, which is the heart stops pumping and you get this excess accumulation of fluid. This happens in humans as well. Congestive heart failure in humans is basically characterized by pericardial fluid edema accumulation, fluid accumulation in other parts of the body as well. And so this is a hallmark of vertebrate cardiac stress. And then just a notation about pHs, and something that's important is that the risk equation for these compounds is different for embryos and small fish than it is for adults. And there's a couple of reasons for that. One is that at the embryonic stage, most of the uptake is across the skin, and the fish haven't yet developed a liver, so they lack the metabolic protection to break a lot of these compounds down. And as a consequence, they accumulate relatively high tissue concentrations, and you see correspondingly severe effects of pH during early development. With the older fish, typically the uptake is via the gills and ingestion, and you're basically running the blood supply through the liver as a measure of hepatic metabolism and protection. And so as a consequence, you get less tissue level accumulation, and you have much more modest pH toxicity. So it's a feature of the size of the embryos and the fact that they don't have a liver, and the fact that they bioconcentrate a lot of these compounds is why they're so much more sensitive to these pHs. And so that's not that pHs aren't issues for other life stages, but a lot of our work is on spawning habitats. As I mentioned, Deepwater Horizon providing opportunities again to break through some of these big uncertainties because of the funding availability. So this is work on bluefin tuna. We've working with Barbara Block's lab here down at Stanford in Monterey. And so Fabian was the lead on a project. This is an isolated heart muscle cell from a bluefin tuna on the end of a glass microelectrode. And what we're doing in this study is we're actually looking at the effects of these PAHs on the ion channels that drive the excitable properties of that cell. So you see the heart muscle cell, when it's been removed from the heart, it's still beating. So it's going through these rhythmic cyclical contractions, right? And those contractions are controlled by the flux or the movement of ions across very specific channels in the heart muscle cell membrane. And so that's shown here. And this is a very, very simplified diagram. But at the top, what we're showing essentially is that the PAHs are acting like drugs, like pharmacological agents. And they're interacting with these and inhibiting these channels, these potassium channels, these sodium channels, and calcium channels, right? And it's this process where the cyclical contraction of a heart muscle cell involves the influx of sodium and calcium, which is shown here on the action potential. So sodium influx, calcium influx, that gives you the upswing and the action potential. And then you get the efflux of the outflow of these potassium ions that bring the action potential back down again. So it's essentially this wave of basically charged ions moving across the membrane. Calcium coming into to the inside of the muscle cell releases more calcium from internal stores via the ryanodine receptor. That calcium then triggers the contraction of the muscle filaments. And then everything has to reset. You got to pump all the calcium back into the ryanodine, into the endoplasmic reticulum, and then reset all of your ions, and then start the process over again. So ultimately, this is what a normal action potential looks like as a function of those flows of ions. And then at the heart level, the tissue level, this is how this translates into a waveform, an electrocardiogram or an EKG that you would get when you go to see the doctor. And again, this is what happens with these muscle cells on oil. So essentially, if you shock or give an electrical stimulation to an isolated muscle cell, you get exactly the same action potential waveform that I just showed you over and over again. You do 1,000 times and you get complete superimposition. But when you start to expose these muscle cells to PAHs, you get this elongation of the action potential, and then essentially it can't repolarize, and the muscle cell goes haywire. And so what's happening is that those potassium channels that bring the waveform back down again are getting blocked by these PAHs, right? And this is a really big deal because the embryonic heart, as it develops, essentially these normal properties of contraction and blood flow are themselves driving the normal development of the heart. So there's this form and function relationship that if you perturb this really delicate balance, there's really long-term consequences for the fish. Okay, back to Puget Sound. What does this all mean for us for monitoring? So this is an old map. It's being updated by the partnership in in tandem with ecology, but essentially there's just a ton of stormwater outfalls. We don't actually know where they all are. And a lot of this information is really old because these conveyance systems were built decades ago. But suffice it to say, where there are people in Puget Sound, there are a lot of stormwater outfalls. Some of them go into rivers that are supporting salmon, some go directly into nearshore areas. And in those nearshore areas, of course, we are concerned about the health and longevity of forage fish, particularly surf smelt and Pacific herring and sand lance. We've done a tremendous amount of work on Pacific herring, again, because that was one of the model species from the Exxon Valdez study work. This is an example of a spawning event off Bainbridge Island. And essentially when this happens, you can see just the aggregation of these spawning adults and then the release of eggs and sperm as this big kind of white cloud. So they're right in those areas where, during a storm event, like in the Southern California Bight, when it rains and you have satellite maps, you can see land-based runoff and sediment into the entire California Bight. So around here with our rain events, particularly in the fall or in the summertime when you get a big storm, you're going to get a lot of discharge into shoreline habitat areas. And again, this ties into the Puget Sound Partnership and the vital signs process with the forage fish. Okay, so back to phenanthrene again. What I'm showing you here is, again, this chromatogram, or sorry, this histogram that shows the list of pHs with the naphthalenes, the 2-ring compounds on the left, all the way up to the 5-ring compounds on the right. In stormwater, just like in crude oil, you see a concentration or relative high proportion of these cardiotoxic phenanthrenes. Those that are in stormwater go right into fish embryos, into herring embryos. And this is what a normal heartbeat looks like in a herring embryo. So there's, again, 2 chambers, the atrium and the ventricle. And I just kind of want to impress upon you, this is the phenotype. So this is normal. And what you're seeing is a 1-to-1 beat atrium to ventricle. And look at the anatomy. At this point in time, what happens is that as the heart develops, the ventricle is beginning to grow out in a posterior direction, right? So I'm going to talk a lot about this particular process. You're going to see a lot of figures that have me pointing to this anatomical feature, this outgrowth of the ventricle. This is herring that's been exposed to pHs, right? So the first thing is the heart slows down. This is a bradycardia. You still get chamber formation, so the atrium and the ventricle. But what you see here, in addition to a slow heartbeat, is you see that the ventricle is not developing properly, right? So you get this kind of stubby posterior end, and then you get the canonical, like in an oil spill, accumulation of fluid. So here, you're right in front of the yolk sac, and here in the pericardial space, you've got this big accumulation of clear fluid. And so if I didn't tell you that this was a stormwater exposure, and I showed you a herring that had been exposed to North Slope crude oil from Prince William Sound, you wouldn't be able to tell the difference, right? So from the herring standpoint, this level of phenotypic injury is well known to us, and it really does back up this thing. Like every time it rains in Puget Sound, you have an oil spill of potentially, a very, very significant amount of pHs that are coming from non-point source pollutions off our roadways into spawning habitats. And the fish are telling us that from a cardiac standpoint, it looks just like an oil spill in Prince William Sound. Oh, and I should mention this particular project was spearheaded by Louisa Harding. It's probably known to a bunch of you. She was a postdoc between my group and Jen McIntyre's group, and is now doing great work with the WDF&W T-BIOS focus group. Okay, so for the past decade plus, we've been really focusing on what's going on with the fish that are not acutely killed by oil exposure, right? So at high concentrations, or if you can see oil on a beach, that level of exposure is going to kill pretty much all of the embryos that are anywhere near there. So what happens is that the space of an oil spill in time, the impact, or of a stormwater discharge, right? How much pHs get in the environment over what period of time and how far they go when they're released, for example, on near shore. All of that, essentially is the impact footprint if the fish are responsive. And so we're really looking at very, very, very low exposure concentrations, trying to understand sublethal effects, not the fish that are acutely wiped out, but what are the more subtle effects on delayed mortality that bring into play ecological death, right? Because we know if we take fish and we put them in pHs, they will show abnormal heartbeats, like I showed you, but then they will recover. If you put them back in clean water, you'll see some restorative potential, but they don't come back normally. They're compromised, and the reason for that is that there's this form-function relationship that I mentioned earlier, where you're trying to essentially build the plane as you're flying it. And so if you remodel certain aspects of early development, then you're going to have permanent changes to the shape of the heart that are going to affect performance later in life. And for forage fish, you know, cardiac performance is everything for herring in terms of their ability to avoid predators and eat. So the fish later, ecological death means they're either going to starve or they're going to be eaten. And so that's the model for linking long-term delayed ecological consequences of short-term trace exposures. And again, these studies that are looking at delayed effects are really focusing on the ventricle, as I mentioned, and the outreach and the extension of that posterior chamber. And the way that works essentially is you have this really complicated developmental process where you start adding cells through proliferation and then migration to create that big complex. You know, you're going from a simple tube to a posterior ventricle extension and to a muscle heavy subadult ventricle that is doing the heavy lifting of the heart. And what we see, again, in a normal fish here on the left, 3 days post-hatching, 9 days post-hatching, and then 4 months later, these are herring, is that if you give a transient exposure to pHs during early development and put them back in clean water, as I mentioned, the heart's going to survive. But if you see the fish on the right, those surviving fish in clean water are showing developmental effects on that ventricular growth. And as I also mentioned, when you look 4 months later, it looks like the fish caught up because now you get basically this big, huge ventricle. But if you look more closely and section that ventricle, this is staying with an antibody to light chain heavy myosin. And what you see here is the number of cells in the ventricle plane versus the amount of area covered. And this is a classic indication of what we call ventricular hypertrophy. So what's happening is that you get this aberrant compensatory response to an early developmental insult that causes hypertrophy in the ventricle later in life. And this is a permanent effect. And what's happening is that this proliferation of tissue is clogging up the interior spongy myocardium. And this is the business end of the ventricle, right? So this part of the heart essentially is structurally malformed. Then the consequences for cardiac output and swimming performance are direct. And we've shown that in multiple studies. Okay, so this is the new stuff that's of interest to the monitoring community. So what we're trying to do is develop a suite of molecular indicators. These are genes that are turned on or off in relation to these injury phenomena that I'm talking about that are anchored to cardiac function and basically reflect environmental exposure conditions in fish habitats throughout the West Coast where there are pHs, which is everywhere there are people, and then ultimately trying to relate that in a way to the so what questions. Why does this really matter for fish and fish health, right? And again, as I mentioned, we're going to really focus on the ventricle, posterior ventricular extension and cardiac morphogenesis. And we think that this process that we're studying underpins a big mystery that was an outcome of the Exxon Valdez analysis. And our colleagues that worked at NOAA at the OCTAVE lab up there did some absolutely Herculean studies where they exposed pink salmon embryos to trace concentrations of pHs, put them in clean water, raised the fish up in net pens until they looked perfectly normal, took the ones that all looked healthy, let them go, and then looked for effects on marine survival. And what they found was a significant relationship between relatively trace exposures to pHs and decreased marine survival. And we think we now know why. We think it's that these fish that look outwardly normal, that have, if you just look at the heart of that oil-exposed fish from the outside, it looks normal. You wouldn't be able to see the hypertrophy until you cut it open and did the antibody staining. So we think that this is retroactively the reason why this subtle effect at a population scale was observed in Prince William Sound 30 years ago. And when I say Herculean studies, Ron Heinz and these guys, they coded wire tagged hundreds of thousands of pink salmon for these releases and did that experiment multiple times. So again, this represents decades of NOAA kind of investments in this topic area. The major goal is biological scaling. We do this all the time. I picked an example from another context that we work on, the pesticides and endangered species. But you know, in ecotoxicology, we're in this dynamic between, particularly for NOAA, where we're trying to address major conservation issues, right? So as I mentioned earlier, our statutes like the Endangered Species Act, we're managing salmon at the population scale or the ESC scale, and so ultimately we're really trying to make decisions at really high biological scales of the population. But much of the work that we do, the biology we do, as I mentioned, is affecting things like ion channels and receptors. And so it's very molecular and it's very cellular. And you know, at the cellular molecular level, 9 times out of 10, some kind of a stress is not necessarily going to be that meaningful. It's not going to translate into an adverse outcome. And ultimately, what the goal is to identify those interactions that are meaningful, like 6-PPD, and then essentially build a conceptual understanding of how those effects at the molecular level cascade through, you know, cells and tissues and into organs and into the whole animal in ways that are going to be predictive of individual survival and fitness, and then from there to population productivity. And so we have this major dot connecting exercise that we're constantly doing. And what I'm going to show you is what this kind of scale looks like, this biological scaling for pHs. I'm just going to turn this thing on its side, right? So just imagine this whole thing flips from left to right. And so now the molecular biology is on your left and the organismal level outcomes are on your right. This is what we call an adverse outcome pathway. In ecotoxicology, the stressors over here on the left, these are these tricyclic pHs, including phenanthrene. MIE stands for molecular initiating event. And there's 2 major pathways right off the bat. One is a subset of compounds are blocking that potassium current and causing the waveform elongation that I mentioned. The other set pathway is blocking the calcium handling itself. And so you get weaker muscle contractions because there's not enough calcium that's doing its job. Those molecular interactions at the receptor level propagate to the cellular organ. KE is key event at the cellular level, key event at the organ level, another organ level event, all the way up to the individual animal, right? To the right, organismal. The reason that this is hard but important is that if you can demonstrably show that one of these events at this biological scale is going to lead to an adverse outcome at the organismal scale, you can use that particular marker, and if it's a genetic marker, with some confidence in monitoring studies, right? So ultimately, if you see a change in the megregulation of these, say, myocardial genes in a really definitive way that's defensible, if you see up or down regulation of those genes in fish embryos, in the field, in habitats where pHs are driving cardiac toxicity, you can have some confidence just from that gene metric that the outcome for those fish is going to be actionable from the management standpoint for NOAA Fisheries. So that's the brass ring. And I should mention, and I'm not talking about it today because we don't have time, that we are in the publishing multiple transcriptional papers that look at a ton of genes in this pathway. I'm only showing a few from our past papers, but there's going to be a tsunami of new information, and the toolbox is blown up on this in FY24 in the coming year. And I just want to make the case for this, right? This is 21st century, right? 20th century monitoring was go collect the fish and measure whatever chemicals are available to you through your analytical techniques to estimate body burdens. Go to the literature for toxicity studies and try to say something about whether or not those measured levels in the environment chemically are going to have some adverse effect on your fish. Most of the work that we did in the Lower Columbia for years on PCBs or PBDEs or pHs and dietary exposure in juvenile salmon, all of that work fit that model, right? But essentially your assessment capability is only as good as your analytical chemistry, right? So if you don't have a method or you don't know about a compound like 6-PPD, you don't know what you don't know. And in many respects, the chemicals that we're measuring, the sensitivity of those instruments are really important in terms of false negatives, right? So what I'm showing you here in this study, this is a paper that just got accepted yesterday. So it's going to be probably hitting the press in the next couple of weeks, is what we've done is we've run the exposures for herring so low that we actually can't reliably measure differences in the levels of pHs with our conventional analytical. So these are pHs in water at day 1 and day 10. And essentially these exposure levels, 0, 0.25, and 5, these are loadings of gravel into our gravel columns. We're below the level of quantitation for GCMS, analytical chemistry. Same thing in embryos, right? We don't see a dose-dependent increase in pH exposures by conventional analytical chemistry. However, we see these effects on the fish heart. So these fish that are exposed to basically undetectable levels of pHs are still showing us this phenotype, which is this ventricular outgrowth phenotype. And CYP1A, which is a classic biomarker of basically exposure to pHs, gives us a very nice dose-dependent response to these oils at these trace concentrations. That means that the gene marker is more sensitive to the analytical chemistry, and both the gene marker and the fish are telling us that there are toxic levels of pHs in fish habitats at concentrations below levels that we conventionally measure them, both for oil spills and for stormwater runoff. So this is going to be a bigger deal probably for fish health in space and time than we have previously appreciated. Just like 6 ppd was a bigger deal for salmon habitats on the West Coast, because we didn't have the technology to sort it out. So to close, looking forward, and again, focusing back on salmon, we're really going into the next 5 years looking for ways to integrate multiple habitat stressors, right? If there's 5,000 compounds in stormwater, there's no way we're going to be able, we just don't have the resources to break them all apart and characterize each one of them. So, and at the same time, we've got these other habitat stressors that we're increasingly having to deal with that are not chemical, so and particularly related to climate change. So we're going to spend a lot of time in the coming years really focusing on the heart as an integrative physiological framework to look at different types of urban growth impacts mediated by poster child compounds, including 6 ppd and pHs in combination with these other habitat stressors, including surface water temperature, which we know has major implications for heart function in fish. And so essentially with the 6 ppd mechanism, you can see these fish are in respiratory distress. It is a hallmark of the syndrome, the urban runoff syndrome. These fish are gaping and swimming at the surface. So more work to come on that. We're going to be doing mechanistic studies, and others are asking the same questions this winter. I just showed you a bunch of data about how pHs are cardiotoxic. And again, people have been studying interactions between thermal stress and cardiac function in salmonids for years. And so our goal in the future is to build an atlas of normal heart development so that we can then look for perturbations as a function of converging urbanization and climate change at the level of the heart. And then use next generation big data, molecular biology, to identify tools, gene probes that can be used by the community for all the monitoring that I talked about in the recovery domain, the regulatory domain, and the habitat restoration domain. So it's a big challenge ahead for us. Team's pretty excited about it, and looking forward to future updates.
Awesome. Thanks, Nat. All right, everybody, we are opening the floor for questions. You guys can unmute yourselves, or you can type stuff in the chat. I can see the chat right now, so I can read stuff off. Dawn, go ahead.
Yeah, that was fascinating. So I coordinate the monitoring work group for salmonids in the Puget Sound for PNAMP, and also work for a company, a firm that does a lot of local land use planning with jurisdictions all across across the state. And I guess my question is, have we established, and pardon if this is like a newbie question, but have we established if those stormwater ponds and sand gravel filters that are so good at getting 6 ppd quinone out, are they as effective on PAHs?
I'm going to leave the question about the detention ponds. So there's a lot of gray infrastructure associated with the extant transportation grid that is mostly built around water volume. And if you go back to the literature of stormwater and kind of who's been leading in this area, it's been the engineers for decades, right? So the engineers are predominantly concerned about safety, public safety, water flows, and flooding control. So most of the detention ponds that are in place along I-5 and elsewhere are meant to hold water and then slowly release it so that you don't have the hydrologic scour and flooding and problems and the like. That process works for water quantity, but it doesn't do much for water quality, right? For a lot of these compounds that are relatively persistent in the dissolve phase. To your other question about whether filtration or other mitigation methods work, when we did the soil column studies, and we've done a bunch of them, and those are still ongoing now, we actually hadn't yet identified 6 ppd quinone. So we didn't know what was killing the salmonids. And if you look at our papers, where we're doing essentially highway runoff from the 520 bridge in Seattle, and then running it through these experimental 60-40 soil columns with soil and compost and sand, they're very effective at protecting against acute mortality, which we think is a 6 ppd quinone, and we're verifying that. And then secondarily, we were measuring pH removal, and the pH levels went way down also. So it's likely that the green infrastructure methods that are out there are going to be very effective at capturing multiple classes of contaminants, we hope, And again, using the fish as indicators when you don't really understand the nature of the stress because you haven't characterized all the chemicals, the fish are telling us in the receiving waters, at least in these bench tests, that they're much happier. But the problem is that these fish are highly migratory, and anything that's going to work in terms of stormwater mitigation has to work at the watershed scale. So it's very much analogous to how much large woody debris or how many stream restoration projects that are physical, how many do you have to have longitudinally within a river system to have healthy restoration? It's the same issue with stormwater outfalls, except for you're not putting wood in the river, you're removing stormwater contaminants from the discharge. So, but conceptually, the challenge spatially from a landscape analysis is very similar.
So, but could the nexus not be looking at these transportation plans and infrastructure plans with their each of the jurisdictions and getting them to start moving in the direction of filtering all of the stormwater before it comes out of either ponds? Or, I mean, I know it's a huge monumental task, but you got to start somewhere, right?
Well,
and
there's
start incorporating that.
Yeah, sorry, there's two ways to do it. One is source control. And so
Yeah.
if you've tracked our work in the past, we did a bunch of work on metals in vehicle brake pads in the 2000s, and that went to the legislature and helped inform the passage of brake pad legislation in Washington and California. That led to the phase out of copper and zinc and nickel and lead and some other metals gradually nationally. That's a situation where there were alternatives like ceramic brake pads or other materials that are not releasing heavy metals, and the brake pad manufacturers agreed to phase out to reduce metal loadings. We can't do that with 6-PPD right now. So the 6-PPD in tires is a structural additive that keeps the tires from blowing apart under heavy friction and ozonation. The tire industry is estimating it's going to be years and potentially longer before they can have a safe alternative for 6-PPD in tires. So because of that, at least in the near term, from a salmon conservation standpoint, we're going to have to tackle this problem by intercepting stormwater runoff from roadways and treating it with green infrastructure. So it's going to be the Wild West of a lot of adaptive, exploratory activity. And there's no one size fits all, right? Because some of these highways are in areas where there's nowhere to do a wetland to treat this. There's no way to do infiltration because you got the wrong kind of soil, et cetera. So there's engineered solutions. There's naturally beaver ponds are being talked about as essentially a big strategy potentially to remove and filter pollutants. But I will say that the federal task force, federal highways, EPA, NOAA, others are working to try to pool our resources to identify common treatment strategies for roadways and other things that are going to be transferable, not just in Puget Sound, but throughout Washington State, throughout the Pacific Northwest.
Thanks so much.
Awesome. Great question, Don. Brian, I see your hand up, and then Whitney, you're next. So Brian Mashoff, go ahead. I see you're unmuted, but I don't hear you. Yeah, still nothing, Brian.
You could type it into the question into the chat for you too, Megan.
Yeah, totally. If you want to try that, Brian, that'd be great. And then I'll just kind of skip over you while you're figuring that out. Whitney, go ahead.
I was wondering who is looking at beaver pond effects on water quality in the Puget Sound?
Not in this regard. We've been talking to Michael Pollack, obviously, in the watershed program. So at our science center with Morgan and I, the watershed program is where most of the beaver pond work has been done. Although Chris Jordan in Oregon has been spearheading collaboratively projects in the interior basin. So there's a lot of folks that are working on beaver ponds in the restoration domain. And they're thinking about water quality in terms of sedimentation and temperature and dissolved oxygen. And so we're hoping to pick demonstration projects in both east and west slope to see how those kinds of mitigation strategies are going to influence basically the water quality upstream of the beaver installation and downstream. And there are other municipalities like the city of Gresham in Portland that is doing some really creative work around green infrastructure involving beaver ponds and the like. And so this is one of those things where I think crowdsourcing and innovation is going to come both from top down and bottom up. And again, it's going to require some novel collaborations with people that have been thinking about physical habitat like beaver restoration and chemical habitat like my group, and kind of putting the peanut butter and the chocolate together. So I'm hopeful that the next round of mapping that we do for vulnerability, if you followed our work, we've produced these predictive maps of where we think fish or coho are going to be killed by stormwater runoff. And the next round of mapping is going to have a temperature component to that. It's going to potentially have a beaver dam component to that. potentially have all of the fish passage planned restoration projects in Puget Sound qualitatively laid over that, so people can start wrapping their heads around where, for a given watershed, the challenge is going to be to deal with these issues. The non-point source pollution is so hard because it is a watershed level problem. And so you really can't solve this with one project. You have to think about them all together, which is very jurisdictionally very challenging. Not for you guys in monitoring. Yeah, I mean, you guys are in this zone all the time, but for a lot of people it is.
I think I might be part of the bottom up part. I'm a citizen surveyor at Longfellow Creek, and I've been doing that for 4 years with Puget Sound Keeper, and recently hooked up with the folks monitoring the beavers there. And I've seen firsthand the expanding beaver dam and also the lowering of the acute impacts. So the salmon run just started, and instead of the first fish coming through and being completely impaired, the first fish coming up looked quite happy. And so just looking, I'm also interested in if there's somebody out there who wants to help test the upstream and downstream water quality and the fish that are coming up. I'm very interested in hooking up with them.
We have more data for Longfellow Creek than any other watershed in Puget Sound. So if that happens, we'd be very interested. And I know the beaver dams you're talking about. I've climbed over them myself.
Mm-hmm.
So yeah, great points.
Awesome. Great question, Whitney. Brian typed his in. Thanks, Brian. Explanation for difference in toxicity of 6-ppd quinone for coho versus chum and other salmonids.
Yeah, that's a great question. It is at the heart of what many of us are trying to do. If you've read our papers, you've seen we've done side-by-side exposures with chum and adult coho. The chum are bulletproof and the coho are incredibly sensitive. We've done further juvenile side-by-side exposures. We published a paper last year looking at sockeye, chum, chinook, coho, and steelhead. And on the gradation, coho are the most sensitive, steelhead are secondary. We see some hints of lethality in chinook, but more work is needed. And we see almost no response from chum or sockeye. That's been borne out across multiple studies. Since this paper was published, since Jenny's paper came out, it's been cited 500 times in 2 years. So the amount of papers that are entering the literature on 6-ppd quinone is relatively unprecedented for my field for ecotox. And it involves fish, because now the stuff's been synthesized and anybody can buy it from Cigna or another source and then do LC50 type studies. So we're getting some good data and a lot of bad data from fish species from all over the world. So it looks like there is among the salmonids this gradation of sensitivity, and it's likely related to the mechanism, right? So when I showed you guys that we did all that work with Stanford to identify the potassium channels as the molecular target for phenanthrene, that's what we need to do for 6-ppd quinone, and it's these kinds of assessments are challenging. Physiologically, they're hard to do. So, but there's a lot of people that are working on this right now that are really good. And as a government lab, we're not like academics or a private company where it's on us to make that discovery. If some other smart group comes along and figures this out, then so much the better for us, because we're just trying to plug it into the management stream for NOAA. But we also, because we have so much experience in this area, are planning to hit the mechanism question hard this winter. And ideally, you'd identify it first in coho and figure out why coho are dying, what 6-ppd is interacting with. It's that cardiorespiratory phenotype that I showed you, right? That the fish are gaping and gasping at the surface, right? So the lead target organs are going to be focusing on the gill and something wrong with respiratory uptake and gas exchange. Another is going to be cardiac function, and the third is going to be a neurological and neurobehavioral component, or all three. And so we'll have, I hope, something to share with you guys by the spring, because we're going to hit that question hard this winter. But essentially, once we figure out the mechanism and why coho are so sensitive, that will give us traction to begin to look at that same process in these other species and hopefully identify why there's such a strong species-specific difference. I didn't believe it when we first started out until we saw this in 2008 with an overlapping chum and coho run in Piper's Creek in Northwest Seattle. And in that year, we had a big chum run come on top of our coho, and all the coho in that system died that year, and all the chum survived. So that was the first field evidence from me personally that I was like, no, this is real. But we don't know why. We're working on it. It's a great question.
Excellent. All right, Casey, Justice, go ahead.
Thank you. Amazing presentation. I'm curious if there's any evidence that adult salmon that are migrating through like a more populated lower portion of the watershed and are exposed to these toxins are transferring any impact from those to their embryo offspring if they successfully spawn higher up in the drainage where there's less toxins?
Yeah, the answer to that is yes. And it depends on the class of compounds. So classic kind of ecotoxicology 101 are these hydrophobic compounds like DDTs that accumulate in fat, so they're hydrophobic. So those are the ones that get transferred up the food web. And that's why they're a concern for southern resident killer whales, for example. So classes of compounds in that category are PCBs and the DDTs and the PBDEs, the brominated fire retardants. It doesn't look like neither the pHs nor the 6-PPDs are particularly showing evidence of bioaccumulating or bioconcentrating in the food web. But the maternal transfer in fat from female vertebrates, be it from killer whales that are offloading a lot of contaminants to their offspring, or to salmon that are offloading a lot of these same contaminants into the embryos, that process is happening. And there's a lot of good thinking about that, not so much in urban watersheds, but in the LWA, because in these areas where you're restoring otherwise pristine habitats, there's a lot of good questions about whether marine-derived nutrients are going to come along with marine-derived contaminants, right? Both in the carcasses of these fish that remain and also as transferred to the next generation. So it's a good question. It is definitely happening. It's hard to resolve in heavily polluted habitats like urbanized watersheds. And I think you'll probably get the best evidence for that in Puget Sound, actually from monitoring in the LWA.
Thank you. That makes a lot of sense.
All right. Excellent, Casey. Anybody else? I don't see anyone with hands raised. Chat.
John has a comment, a question in the chat. Dawn Spilsbury, who we heard from earlier. She says, coho spend more time and perhaps more contaminant exposure in freshwater natal areas prior to outmigration than chum, then coming back as compromised adults and more sensitive, maybe. Sorry if I butchered your comment there, Dawn.
No,
just
it's a great
thought, I think, is what she's poking.
Sorry, go ahead, Megan.
No, go ahead.
No, this issue about life history and risk among different salmonids, even within a species, right? Like you're talking about differences between chum and pinks, right? Those fish are doing relatively well, and what they have in common is they don't hang out in fresh water where water quality and pollution and non-point source runoff are really a big deal. And the fish that do spend time in freshwater for longer periods of time, the steelhead, the chinook, and the coho, are really in trouble, right? So it could be coincidence, but I don't think so. And it's not just a water quality problem, it's just land use problem, right? So water quality is bad when you have these other problems related to human activities that affect flow and, you know, the introduction of non-native species. I mean, that's why the urban stream syndrome is so hard to crack, because it's an all of the above problem with humans impacting watershed function. But with respect to this issue of prior experience, that was a big thing for Longfellow Creek, right? For these salmon that were coming back and dying. Early on, there were questions about whether the fish that were born there were somehow compromised in a way that set them up for failure later in life. Most of the salmon that come back to Longfellow Creek, however, we know this from pit tags and the coded wire tag analysis that we've done, don't come from Longfellow Creek. They're straying from the Muckleshoot net pen operation. And that's true for a lot of our urban streams, that if we didn't have artificial propagation from the hatchery operations, many of which are tribal, we wouldn't have any fish in our urban watersheds at all. And we wouldn't have been able to do the research that we did to actually discover this habitat factor that's been under our noses for 50 years. So it's not that those fish are set up or preconditioned by a prior exposure. It's an acute stormwater runoff. And with pHs and 6-PPD, it really is. If the fish is there in the wrong place at the wrong time, it's going to be hit by whatever's in the runoff.
Ooh, excellent.
Yeah.
And Meg, there's one more comment in the chat from Brian, just a follow-up comment regarding potential point source impacts of artificial turf soccer fields. And I know from my experience in our community that shredded tires are specifically used in turf for all kinds of sports fields and playgrounds, and you can buy it for your backyard now. So I think Brian's pointing out that maybe that's a potential point source as well to consider.
It is, and it's talked about frequently. And I never thought in my wildest days of being in grad school that I'd ever be thinking about soccer field runoff into salmon habitats. But it is the tire materials themselves. The tire chemistry is complicated when you shred them, when you handle them in different ways. So, you know, if you're a researcher working on tire-related compounds, the tire industry is going to have a lot to say about how you make artificial tire particles or tire wear or tire tread. So I just want to be careful to say that how they shred those tires and how old those tires were and where they came from will affect what's still in those tires, but they're still tires, right? So if you soak them, if you take tire dust from your installation of interest and you soak it, there are tools to let the fish tell you whether or not they're toxic, and they probably will be.
All right.
Thanks, Nat.
Those are some great questions, guys.
Yeah, thanks. I appreciate that.
Anybody else? All right. And that
Wait,
anybody,
there is one more, Meg.
ooh,
Megan Wilson from Puget Sound Partnership asks, what type of research or resources would be needed to develop the genetic indicator of cardiotoxicity you described for the Puget Sound?
That's a great question. This is one of the task areas that's under the new 4-year agreement that we have under the NEP with EPA Region 10. So we are already putting the resources that we have through NOAA and through EPA joint agreement. And it's also, NOAA was relatively late with IRA funding, but the package that is being considered internal to NOAA Fisheries right now includes, and this is still provisional, so it's not a final determination, but there is a request that is likely to get funded for the equipment that we need to identify and localize these genes that I'm talking about with the papers that are forthcoming into the developing part. And the goal there is to validate those biomarkers. And so we are already heading in that direction. It's not like an unfilled request. And so the good news is that, and you know, 5 years ago, it would have been a wasteland for funding for this. But right now, with these investments in the transportation grid through BIL, and in climate and transportation through the IRA, the federal agencies know that if we don't get out ahead of the SCAP, because remember, in Puget Sound, the investments that are ongoing right now are once in a generation. There's more money going into the transportation grid than at any time in recent memory. And that money, those investments will build the skeleton, the framework for where we grow and add people to the landscape for decades to come. So we have an opportunity to get this right or wrong with this non-point source pollution and stormwater thing. And it's going to be a really big deal with respect to IRA investments in habitat restoration and fish passage, because we don't want poor water quality to create ecological traps upstream of culvert removal projects. So the federal family is really activated in this area and focusing on sharing resources. And again, to that point, the cardiac markers are really badly needed because we want to do all this monitoring to make sure that whatever investments are made are working the way we want them to and the fish are healthy. So we're under the gun, and there's more money for this than there has been. And so we'll have more to report, I hope, by next summer.
all right, excellent. Okay. Megan says thanks.
Thank you.
Okay. All right, awesome. I think we have about 5-ish minutes left to wrap up. So I'm gonna do some wrap ups, and then Nat, is it okay if I distribute your email address if anybody thinks of questions after the meeting or comes up with anything and wants to kind of pick your brain later? Would that be okay to include your information?
Please do. And as I mentioned, this is a really dynamic research area. And so feedback from this community is really important to us, right? Across the board, and not just for Puget Sound, because our area is the entire Pacific Northwest, the West Coast. So if anything resonates or you have ideas or questions or whatever, please do reach out. We appreciate it.
Excellent. Awesome. Thank you so much, Nat. That was an awesome presentation.
Thank you. I'm signing off then.
All right. Thanks, Nat. All right, and I will take control of the screen. Myself resituated here. All right, everybody. Thanks for sticking around. I will wrap up with a reminder that some general, just with some general fish monitoring workgroup information. You can always access more information by visiting the project page on pnamp.org. Jen can drop that link again. The project page contains links to each task page where you can find task descriptions, documents, upcoming meetings, links to previous meetings, or ways to contact the task leads. If you'd like to be added to the distribution list, just send me an email. I'll be sending out a survey later to gather feedback on how we can improve on or things that you would like to see in 2024. If there's a particular topic you'd like to learn about, if there's a particular speaker you would like us to try to wrangle, just let me know, and that'll go out with the follow-up email. The recording of this meeting will be available next week. This group will convene again on January 18th from 10 to 11:30. And to close out for today, I would like to give a big thank you to Nat Scholz for presenting, and to our core team, task leads, and all you guys. Thank you and have a great Thursday.